briefly describe the procedure that we adopted for the selection process. And this is a procedure that we as a committee decided upon together after some brainstorming sessions. There was basically two stages. In the first stage, a large number of researchers were asked to nominate papers, whatever they felt they would like to see in the list. And they were allowed to nominate their own papers as well, as long as they also nominated others' papers. This is kind of an incentive to get them to actually do something, go online. So that way we had a list of nominated papers, all of which were put together in an online poll, which was then made available to a more selected group of voters who were all speakers of this conference, both keynotes and selected speakers, as well as all keynote speakers of past versions of this conference. So that was also quite a strong list of people who voted on this in the second stage, and then the papers were sorted by the number of votes they received. Each person was asked to vote for up to 5 papers in the second stage. A short disclaimer, I think we've got a fantastic group of papers coming through the selection, but we do not claim or pretend that this list is perfect, complete, sound, or that the process was foolproof. Don't sue us. This is the first attempt that we made at an award mechanism which I think should serve the community in some ways. We definitely welcome your input for future iterations of this newly instituted award series. With that, I'll just go over to the list of top 10 papers.
I think, and before we begin, I think this is also a good opportunity to bring attention both to this particular selection process for next year's. This way we actually have more input and participation, not just from the folks in this room, but from the wider community. And I think that we're going to have an opportunity to post this in our website, in the website of the ISCB SIG, in possibly bioinformatics as part of the summary of this meeting. So I think these papers will draw some attention, but what we're really hoping is that this is a first year and a seed to have a more formal process and to continue getting outstanding papers for this. I mean, I have to admit that both of us were completely frightened when we started doing this. We're like, what if we only get biased sampling and everybody's voting for their own paper, and what if this and what if that? And now we're looking at this list of papers, and we really feel that it really represents our community, and it really represents the way that our field is going, and we're hoping that it will continue to improve in future years. So
yes, so I'll just go ahead and announce the papers in alphabetical order, and I'd request you to hold your applause until or maybe, how do we? All right, so one slide at a time, one paper at a time.
Let me also make a small modification. If you're an author of one of these papers, how about right before the applause, you stand up and you say yay? So take it away.
Okay, so the first one is a high resolution of mass. Human evolutionary constraint using 29 mammals. Lindblad, Toh et al., last author, Manolis Kellis. This paper appeared in Nature 2011.
This one was planted.
Accurate inference of transcription factor binding from DNA sequence and chromatin accessibility data. Roger Pick-Reggie et al., last author, Jonathan Pritchard.
And Yoav Gilad.
Yoav Gilad. Yoav, can you say yay?
We happen to have both the first and the last author here, so do you guys want to stand up and say hi?
Great. An extensive microRNA-mediated network of RNA-RNA interactions regulates established oncogenic pathways in glioblastoma. Sumazin et al., last author, Andrea Califano. Appeared in Cell. Analysis of genomic variation in non-coding elements using population-scale sequencing data from the 1000 Genomes Project. Mu et al., last author Gerstein, Nucleic Acids Research. Cofactor binding evokes latent differences in DNA binding specificity between Hox proteins. Slattery et al., last author Richard Mann, appeared in Cell. I think we have Remo Rose as the co-author.
Awesome.
Comprehensive genome-wide protein-DNA interactions detected at single nucleotide resolution, Re and Pew, cell. Discriminative prediction of mammalian enhancers from DNA sequence, Lee, Karchin, and Beer. We have Michael Beer here. Appeared in Genome Research. Dynamic exchange at regulatory elements during chromatin remodeling underlies assisted loading mechanism. Voss, first author, last author Hager, appeared in Cell. Mapping and analysis of chromatin state dynamics in 9 human cell types. Jason Ernst, first author, last author Bernstein, appeared in Nature. Jason.
Go, Jason.
Prioritizing candidate disease genes by network-based boosting of genome-wide association data. Lee et al., last author Mark Hott, Genome Research. And that's a list of top 10 for 2011. Congratulations once again to all the award recipients.
Just one moment. I mean, these are sorted now by PubMed ID. I mean, we're going to post these on the website. You can also see the journals that are sort of reflecting our community. You see Nature, Genome Research, Nucleic Acids Research, Cell. There were several additional nominations from these journals as well as other journals. So I think we're going to also let the journal editors know so that, again, we sort of reach the wider audience. You also see the number of citations. I think one of the advantages of doing it late in the year is that these papers have had a chance to build some citations. So I think these further sort of put the stamp of the larger community approval to the community that you guys are a part of that actually selected these papers. So let's give a big round of applause to all of these first authors, last authors, and all the authors and all the papers.